Today's session is hosted by NICE, who is one of the ERA4TV partners and who helped within the coordination of ERA4TV external communication and engagement. My name is Alberto Borovia. I am clinical pharmacologist. I am the head of the clinical pharmacology department, the clinical trial unit at La Paz University Hospital in Madrid, Spain. I also the leader of one of the work packages in ERA4TV, the work package 7, that is dedicated to the development of first-in-human trial in the pipeline on ERA4TV. Today, I will be moderating this first session of the challenges to tuberculosis drug development and access. The aim of this webinar is to understand the current and future challenges to tuberculosis drug development and regulation to serve as a first step to moving forward. Next slide, please. Before starting, a reminder to everyone that this session will be recorded and only the speakers will have their cameras and microphones on. Everyone else will be turned off to avoid any disturbance. If you have any questions, please write them down on the chat and the speaker will address them at the end of the presentation. Finally, feel free to post about this webinar on social media using the hashtag provided in the slides, ERA4TV and WAAW 2022. So first of all, please next slide. We will start with an introduction of the ERA4TV project to give everyone an overview of the focus and objective of the project. ERA4TV is a public-private initiative of over 30 partners devoted to accelerating the development of new treatment regimens for tuberculosis. This project was funded by the Innovative Medicine Initiative, IMI, and was started in 2020. It has a length of 6 years and is planned to run until 2026. ERA4TV focuses on the earliest stages of drug development, exploring potential new treatment or treatment regimens in cells, in animal models, and up to the first-in-human trials with the final goal of ending the tuberculosis epidemic by 2030. For that, the ERA4TV platform is based on a progression pipeline of a variety of molecules at different stages of development, and currently the project has 2 molecules in first-in-human development and another few in preclinical stage. The next slide, we can say there are ERA4TV partners bases, as you can see, around Europe and also in United States, and gives you an idea of the magnitude of this project. These are all the partners working together in the slide, implementing state-of-the-art tools and methods such as modeling and simulation tools, and exploring also the use of application of artificial intelligence to drive forward the fight against tuberculosis. Next slide, please. Just before kicking off the session, I would like to make an appreciation and highlight that the length of the webinar session has been scheduled to happen during the World Antimicrobial Awareness Week. As you know, the World Antimicrobial Awareness Week is a global initiative that is celebrated annually to improve awareness and understanding of antimicrobial resistance and promote best practice among the public, healthcare stakeholders, and policymakers also, who will play a critical role in reducing the further emergence and spread of antimicrobial resistance. The theme chosen for this year has been Preventing Antimicrobial Resistance Together. The slogan "Antimicrobial Handled with Care" has been used to draw attention to this complex global health problem and how we can bring it under control. And now, without any further delay, we will start with the presentations. First speaker is Francesca Conradi. She is the clinical research study leader in the Susan Clinic at the Seasway Tropic Disease Hospital in Johannesburg, South Africa. She also holds a lecturer post at the Johannesburg University. She has also been a part of the research agenda that brought effective antiretroviral therapy to millions of South Africa living with HIV. Now she is focused on tuberculosis research, especially new treatment strategies for drug-resistant tuberculosis. She sits on the National Clinical Advisory Committee headed by the National Tuberculosis Program and has developed guidelines for bedaquiline eligibility. And right now also provides advice to our consortium ERA4TB throughout this scientific advisory board. Thank you, Francesca, and please go ahead.
Thank you very much, and thank you for inviting me to be part of this forum. Just to be clear, I'm not a scientist, I'm a clinician, and my job is to find and put into programs new treatment for tuberculosis. I want to imagine for a second that we are not designing new TB drugs, but we're designing new cars. Of course, the analogy will fail after a while, but if you look at how you would design a new car, the first thing that you would do is you'd look at who's going to drive this car. A sleek, sexy Tesla probably belongs to an executive in Palo Alto. If you're going to design a people-moving vehicle, like a quantum bus, it's a different audience that you will be appealing to. And what I want to do in my talk is to really talk about who are our end users. The drugs that we are developing, who is going to be the people who actually swallow these drugs? Where do they live? What are their characteristics? Of course, the analogy falls down with the fact that we're also dealing with a host pathogen and a pathogen response, so we can't take the analogy everywhere. But we actually have 10.6 million new customers next year. That's the number of people that we estimate will get active TB disease in the world. And where do they live and what do they look like? So what I'm going to do on the next slide, please, is the topics that I'm going to cover is I'm going to look at the clinical guidelines for the treatment of both drug-sensitive and drug-resistant TB and how they've evolved over my career. I'm going to talk about, I am a South African doctor, so I'm going to talk about the average DRTB patient in South Africa and in India. I'm going to talk about what it's like to conduct clinical trials in tuberculosis. So once we've got a candidate drug, how difficult it is to actually conduct the clinical trials, what the current and the emerging clinical challenges are. Next slide, please. So I'm going to give a brief history, and I think the fact that I can encapsulate the history of the treatment of TB in a programmatic fashion onto one slide really tells us that there hasn't been many advances in the treatment of rifampicin-sensitive TB since about the 70s. So in the 70s, based on some work done by the British Medical Council, a short course of TB treatment was endorsed by the WHO and rolled out throughout the world. This consisted of a 2-month 4-drug intensive phase, HREZ, and 2 and 4 months of a continuation phase of INH and rifampicin. Since then, there have only been variations on this theme. No one has challenged the duration of treatment, the composition of treatment. We've had variations like, for example, can we give the treatment by directly observed therapy Monday to Friday? And the answer is no. We can't. People need to take their treatment every day. And what's this with watching people taking treatment? Directly observed therapy has also gone out the window. In addition to that, another great step forward, and it is just a variation on a theme, is the co-formulation of these medicines into single tablets. So it's basically taking the mixture of a cake and putting it all into one tin. There have been great improvements in the molecular diagnostics of TB. And I always feel when I'm speaking to an audience like this that we live in two different worlds. The world that I live in, and I live in a moderately resourced country, we have access, we diagnose TB almost always by gene expert, which means that right from the beginning, we know whether this will be rifampicin resistant or sensitive. But that technology only is represented in about 2/3 of the world. Many people are diagnosed with TB on the basis of, I know you're all sitting down, but on the basis of microscopy, which gives us very little information about what treatment regimens to choose. But the use of the gene expert has penetrated into many developing countries as it improved our diagnosis, both as a more sensitive tool, but also as a way of deciding on resistance patterns. In the year 2022, there have been the first real updates in the treatment of drug-sensitive TB. The first one is that we can consider the use of a rifapentine-based regimen for 4 months in an adult. While there are still some holes in this data, for example, can it be used in pregnancy and in people with advanced HIV disease? This represents a major step forward. And the implementation of this will now be, can we co-formulate? How can we get this into the hands of people who really need it, i.e. the people who are going to get active TB disease in the next year? So that was based on a study called Study 31, great step forward. The next was looking at a shortened regimen for children. Pediatricians have told me since ever I can remember that the burden of TB disease is much smaller in children, and we can get away with less treatment. But for the first time this has been actually proven in a randomized controlled trial for children, and that trial is called SHINE. So as we can see, the history, if we look at the treatment for the average person with rifampicin-resistant TB, little has changed since I qualified as a doctor. Next slide, please. If we have a look at the treatment of rifampicin-resistant TB, for a long time, the opinion of, or the guidelines that we were given for the treatment of rifampicin-resistant TB was largely based on expert opinion. So what people who had experience in the field, understanding the microbiology of the organism, tinged with a little bit of what I think was just plain fear. We got our first written guidelines for the treatment of drug-resistant TB in 2011. This was followed by a number of WHO guidelines improvements. We can have the next slide, please. And culminated in this regimen recommendation that patients who suffered from rifampicin-resistant TB with or without INH resistance were to be given an injectable agent for 6 to 8 months. In the developed world, this might be given by a port, but in the developing world, this is given by intramuscular injections. So an intramuscular injection in a patient who is cahectic or wasted for 6 to 8 months, aside from the side effects, is a painful exercise for all concerned. It was suggested that we use a later generation quinolone for 18 months, ethionamide for 18 months, and ethionamide gives worse nausea and vomiting than even the earliest stages of pregnancy. And we were asking our patients to take these drugs for 18 months. And in that, we added a really old drug called cycloserine or taururzidone, and alongside that, para-salicylic acid or PAS was given for 18 months. We used ethambutol and PZA for 18 months. And only very recently, in the last 7 to 10 years, did we discover that at least 50% of our patients have phenotypic resistance to both of these drugs. So we were giving drugs that probably didn't work, or worked in didn't work in 50% of the patients. But these were the guidelines that we were given. Then in the next slide, please, we came to the first randomized controlled clinical trial that was conducted in the space of drug-resistant TB. This trial was published by Andrew Nunn et al. It was called Stream Stage 1. And what it showed was that a shorter regimen with only 4 months of injectables and high-dose INH was non-inferior to the current standard of care, which was the course of treatment that I described before. Next slide, please. And then there was a whole plethora of new and repurposed drugs. Bedaquiline is a new drug, as is delaminid and pretominid, that have actually reached market access. There are novel drugs with novel mode of actions, and all of them, to my knowledge, have been approved by most regulatory authorities. Then we had 2 old-fashioned drugs. Clofazimine, a drug that's used for the treatment of leprosy, was added. Makes sense that it works, but what is not noted is that it has a profound effect on the QT interval, so may cause fatal tachyarrhythmias and causes hyperpigmentation of the skin, which might sound like a cosmetic problem, but may be a barrier to adherence. Linezolid, an old ICU drug that we used, is now being repurposed for the treatment of rifampicin-resistant TB. And this ended, if we have a look at the next slide, please, in a categorization of the WHO for drugs for rifampicin-resistant TB. The top 3 drugs are bedaquiline, quinolone, either levo or moxi, and linezolid, and this should be included in the programmatic treatment of all patients with rifampicin-resistant TB. We can add either clofazimine or taururzidone, or depending on toxicity and availability of the drugs. And then we've got a number of drugs that are called category C, which we can add if for some reason a group A or group B drug is not sufficient. As you can note from this, despite the fact that the WHO has endorsed BPAL with moxifloxacin, pretomanid is not even on this list, not even as a category C drug, which I think demonstrates the fact that guidelines are not keeping a pace with the research that's been done. In the next slide, please. Finally, the WHO rapid communication came out this year, in May this year, and it endorsed the combination of 3 or 4 drugs for most patients with rifampicin-resistant TB. So bedaquiline, pretomanid, and linezolid, with or without moxifloxacin, can be used for 6 months for the treatment of rifampicin-resistant TB. This is a huge step forward. This is a mostly tolerable regimen with some side effects, almost all attributable to linezolid. But it's our oft-quoted Minister of Health says it's 23 tablets a week as opposed to 23 tablets a day, which is what we used before. Slightly counterintuitively, the quinolone can be stopped if the patient has resistance to a quinolone. So the worse the TB, the less drugs you get. That, however, is the guideline. Next slide, please. So now I want to turn this on its head and say, now we know what a really good regimen looks like. It's been trialed both in single and double and in randomized studies. WHO has endorsed it. But what does our population that we're going to treat look like? So if we look at the average, and I know generalizations are not useful, but the average patient who acquires a rifampicin-resistant TB in South Africa, first of all, they are young. They are 35 years old. As in all forms of TB, more males get TB than females, and I'm not sure why this is. I think it may be occupational, maybe related to smoking history, or maybe a genetic predisposition. I'm not sure. 45% of our patients that we see are female, and if they're 35 years old on average, many of them are childbearing potential. If we run through the numbers in our head of what this actually means, 10.6 million people get TB every year, of which about 500,000 of those, it's quite a phenomenal amount, get rifampicin-resistant TB. But overall, about 200,000 women who are pregnant acquire TB every year. And the information that we have on how to treat these patients, the actual hard data, can be written on a very small postage stamp. We have no real PK data. All we're worried about is not harming the unborn baby, which is quite a paternalistic viewpoint. In South Africa, 50% of our patients with TB are HIV co-infected. We have a really good antiretroviral program with over 4.5 million people taking antiretrovirals in our program. So one of the major impacts of TB has not been through improved TB treatment, but improvement of HIV care within South Africa. Most people who acquire drug-resistant TB in South Africa live in the cities. They live in overcrowded, impoverished areas. They are often young. They are often the breadwinners for the family. And anyone who takes a breadwinner out of being able to earn money, it has impact on the rest of the family. Substance abuse in this population group of drug-resistant TB patients is not uncommon. The use of both alcohol and other licit or illicit substances is not an uncommon situation. If we have a look on the next slide, please, at the average drug-resistant TB patient, and I've compared my cohort to a cohort in India, the average age is about the same. Again, the same distribution between the genders, but far less HIV infection in India. And I think that that's more representative of the epidemic of TB in Southeast Asia. If we add Africa and Asia together, Africa and Southeast Asia together, 6.6 million people acquire TB in those areas every year. So this is our target market. This is the population group that we need to be focusing on when we're looking at drug development. Next slide, please. So what is it like to conduct a clinical trial in TB? I'm not sure if my camera's on, but you can probably see I've got quite a few gray hairs, because conducting clinical trials in TB is very difficult. The first thing is that almost all persons who acquire TB or need TB prevention are treated by national TB programs. It is a very political disease, and the national TB programs report to the WHO on notifications of active disease, on notifications of co-infection measurements with HIV, TB, and smoking, and have to report on outcomes. So in terms of politically, the national TB programs are the ones who hold the reins, as it were. So what we need to do is look at what a national TB program needs and align our clinical research to fit in with the national strategic goals of the national TB programs. So we can't answer everything, and we are looking at not the social determinants of ill health, but we're looking at what do national TB programs need. They don't need variations on the theme. They need novel thinking, new modes of action that can provide care to most people. Next slide, please. So the next thing is, when we're conducting clinical trials, to stand up to the rigor of scientific review, we need to have a randomization to the current standard of care. And if someone says to me, what's the current standard of care in South Africa for rifampicin-resistant TB? And I'm, as mentioned before, I'm on the National Clinical Advisory Board. I'm not actually sure what it is. So it's a moving target. We're in the process of changing from a short all-oral regimen to BPA-LM. It's a moving target, and when you're going to compare something to the standard of care, BPA-LM has a very high success rate of 90%, and you need to compare to that. If you want to conduct a clinical trial in more than one country, there are hoops and hoops of regulations that you need to go through. I've worked in South Africa all my life, and I think that I understand the regulatory environment in South Africa and what applications need to be put in where. But once you move out of that, if you want a multi-country study that has better generalizability, the hoops of regulations that you have to go through are massive. Many of the potential participants are often very ill with TB itself, because TB is a stigmatized disease and people often will present late for it. So we've got very ill patients, and what you need to do is to separate out what the adverse events associated with the new drug that you're going to try from the adverse events associated, that's an incorrect use of the term, but associated with their comorbidities. And common comorbidities are HIV co-infection and diabetes. The next thing is, it's difficult to enroll patients when you have such a heterogeneous population. This is not a medical term, but sometimes people say they've got a touch of TB. And what that means to them is that they have very minimal changes on their chest X-ray, maybe smear 1+ positive or negative altogether. And people who have advanced disease can have bilateral fibrocavitary disease with 3+ positives. So how do you talk about a response to treatment when the people that we are trying to give the treatment to range from being mildly unwell to very sick? Next slide, please. So as you can hear from what I've shared so far, is that the uptake of innovation in the TB arena is slow. The fact that we are still using the same treatment to treat drug-sensitive TB as was used when I qualified as a doctor many years ago shows that innovation and adoption of new regimens is slow. As I've mentioned often before, there are 10.6 million cases we estimate that will occur in the next year, and most of them occur in Southeast Asia and Africa. The most common attributable factor, and I haven't mentioned this before, but the most common attributable factor to the development of active TB disease is undernourishment. And I appreciate that this is not a webinar about addressing universal access to care and to food particularly. We've got to bear in mind that people who are undernourished have different pharmacokinetics from those who have a normal albumin. So when we're developing drugs, we need to bear that in mind. There's no use telling me that a drug can only be given with an albumin above 30 in South African units, because I hardly ever see patients like that. The other attributable factors are smoking. I wish we could eradicate that. I think we'd get rid of a lot of TB if we could do that. HIV, alcohol abuse, and diabetes. And when we look at the development of new drugs, and this is in no way my area of expertise, we need to look at the cost effectiveness and what national TB programs are prepared to bear. Many of our TB programs have a very low option for what they will accept, and then we have to balance reduction in the duration of treatment with the maybe more expensive drugs. Then the last thing that I want to talk about is that for one of the first times, a very interesting study called Truncate was published by Nick Payton. And well, at least the results were released in the Union Conference. And what this showed is that if you treat patients with appropriate medicines for 8 weeks, and then watch them to see whether they will relapse or return, it is an effective method. And many of our patients are being overtreated. But we cannot, by the WHO guidelines, treat them for any less unless it's done so by accident. So we need to develop some form of stratification for the treatment of tuberculosis. I know it's going to come as a shock to the people on this call, but probably 2/3 of South Africans who get TB don't even get a chest X-ray done. They are treated on the basis of their gene expert result alone. And this development of being able to stratify medicine to a more applicable approach to the burden of disease needs to be done in parallel to drug development. Thank you very much.
Thank you, Francesca. Very interesting presentation. Now, next speaker is Sophie Cooper. She's a senior scientific advisor at the NICE in the UK. NICE, as you know, is the UK's health technology assessment body responsible for ensuring that new medical treatment, devices, and diagnosis are both clinically and cost-effective. Sophie works in NICE Science, Policy and Research Program and has recently led an innovative piece of work alongside the England National Healthcare System, exploring novel payment and evaluation models for antimicrobials. So, Sophie, it's your turn.
Thanks, Alberto, and thanks, Francesca. So we've heard about the challenges to TB drug development from a clinical perspective, and I'm going to talk about the challenges from a more commercial perspective. So I'll talk about why investment in antibiotics for TB, and indeed investment for any type of antimicrobial drug, regardless of the type of infection, is really unattractive for companies at the moment under the current systems that we have in place to set prices and pay companies for making and selling these drugs. And I'll talk about what we've been doing in the UK, or specifically at the moment in England, to try and address the poor pipeline of antimicrobial treatments and incentivize companies to invest in researching and developing new ones. What we've been doing isn't focused on TB. We've been focusing on actually some other WHO designated high priority drug resistant pathogens. But regardless of the type of antimicrobial we look at, the same principles apply. So the approach that we're testing at the moment and we're looking to roll out next year could offer a solution as well to the lack of the issue of a lack of investment in TB drugs. Just two things to note before I get started. I'll be using the term, and I have already, antimicrobials. You'll probably be aware that's more of an umbrella term for antibiotics to treat bacterial infections such as TB, MRSA, etc. But also, it covers other types of treatment like antifungals, for example, for infections such as Aspergillus, or yeast infections like Candida auris. And also to note, this is I'm speaking on behalf of a few of our project partners here today, so it's a collaborative piece of work between NICE but also NHS England and the Department of Health and Social Care, as you can see from the slides. So next slide, please. So, I think everyone knows antimicrobial resistance is a serious global issue. In 2020, I think it was, the WHO included it in a list of its top 13 global health challenges for the next decade. Because already, so many microorganisms that cause so many common diseases around the world, including tuberculosis, but also malaria, gonorrhea, urinary tract infections, chest infections, and ultimately dangerous bloodstream infections, have become resistant to a wide range of medicines, which can arise through many mechanisms, but the main drivers are overuse or inappropriate use. And with global travel being so easy and prevalent, resistant bugs in one country can easily spread to others. So truly a global issue. And of course, without working antimicrobials, routine procedures won't be possible, and they'll be too dangerous to perform. Chemotherapy will be too high risk. You all know all of this. And in severe cases where infections are resistant to many drugs, people are left with few options, which are often toxic, or even no options at all. And I know in tuberculosis, often you have to rely on a combination of several drugs over a long time. In severe drug resistant cases, which aren't very effective and also toxic. So I'm sure the audience is aware of that issue. And worryingly, the pipeline of antimicrobial drugs in development, in particular the pipeline for treating so-called superbugs, is really poor. And you can see that from the stats here on the slide about drugs that are in clinical trials at the moment. It's really lagging behind for antimicrobials compared to other therapeutic areas outside of infectious disease. So if we took just one other therapeutic area as a comparison, cancer, as of 2020, there were 1,800 immuno-oncology drugs in phase 1 to 3, so human trials, compared to only 41 antimicrobials. Now, for some reason, this analysis of pipeline antimicrobial drugs that we often quote as our project, part of our project, excluded TB drugs, but I found another source cited in a report by the WHO, and that suggested only about 13 TB antibiotics were in phase 1 to 3 trials in 2016. The sources are down on the bottom there. And the reason for this is because the high costs of researching and developing antimicrobials just aren't recouped by companies under our current systems. So any new product to the market for these superbugs and really resistant infections is restricted to very specific and tight usage, for example, reserving it as a last resort drug, keeping it under lock and key. And that's very appropriate, prescribing behavior. We call that antimicrobial stewardship. And so stewardship's a word that comes up a lot in this project. And the aim of that good stewardship and that tight usage is to slow down the development of resistance to the new drug. So in the absence of any significant outbreak of this infection that the drug has been launched to treat, the sales could be really low, particularly during the period of market exclusivity, so the patent period, which is when pharmaceutical companies expect to recoup their costs. Add to this the fact that new products tend not to be used due to their high cost compared to generics, meaning even lower sales. And this actually exacerbates the issue of antimicrobial resistance, because over-reliance on only a few drugs creates selection pressures and increases the ability of bugs to evolve resistance to them. Whereas actually we want to be able to offer a range of different antimicrobials as part of what we call cycling stewardship regimens. And for TB, I think the market failure is compounded even more because of this requirement for combinations of drugs, for really drug-resistant strains, making it more expensive and further diluting the already weak commercial incentives. And another interesting stat from the WHO report saying that investment in R&D for TB drugs is at its lowest level since 2008, and only two, I mean, Francesca can probably quote this better than me, but only 2 new antibiotics for multi-drug resistant TB launched in the last 70 years. So next slide, please. That was just kind of a bit of the clinical driver, and just a bit of a history to the work that we're doing here in England and the UK. It's had a long sort of history and lots of national and international attention. So back in 2015, there was a joint working group set up between UK government and industry to address antimicrobial resistance, AMR, if I use the acronym AMR. I'll try not to use acronyms. And in this part of this working group, the principle of doing something different with regards paying and reimbursing antimicrobials was broadly agreed. And then a few years ago in 2018, a report came out from one of the policy research units to our Department of Health and Social Care. And that presented some detailed methods showing exactly how we might achieve a different payment system and evaluation system, and what that meant for the activities that NICE do. And the link to that report is on the slide. In 2019, the UK government gave a firm commitment to testing these new approaches. So then in 2020, NICE and NHS England selected a couple of products to test this approach on in our pilot, ceftazidime, avibactam, and cefiderocol. And these are for treating, as I said, not TB drugs, but infections caused by gram-negative bacteria. 2021 is missing from the slide. There's lots of work behind the scenes there, assessing the evidence, compiling the evidence, developing economic models. And then this year, our specially convened advisory committee met to assess the evidence and make some recommendations, with a huge milestone in July, marking the start of a new payment system for these 2 drugs that were part of the pilot. I must say here that the project and our work doesn't end with these 2 pilot evaluations. We want to implement a long-term routine approach to evaluating and purchasing antimicrobials differently, and I'll come back to that later. Next slide, please. So what is our new approach, and why does the current one not work? I explained kind of why it's unattractive. So what are we proposing to incentivize the market? So under current systems, companies get paid per pack of drug supplied, in simplistic terms. Under this new payment model, the payments made by the NHS to the manufacturer do not depend on the volume of drug supplied. I talked about sales being low due to good stewardship, meaning they're not recouping their costs. So we're delinking amount paid from volumes sold. And instead, companies are guaranteed to receive a fixed annual fee regardless of how many prescriptions are issued. So that overcomes this issue of strict stewardship, which is good, which leads to low sales. And instead, payments are based on the benefit that the antimicrobial offers to patients and the NHS. And that's where NICE comes in. So our evaluations estimate that value, which translates to the payment. Obviously, we need to get the balance right between rewarding companies for developing great drugs, but not overspending taxpayer money. NICE's estimates of value informed then commercial discussions between NHS England and the companies to agree the final fixed fee payments. That's confidential, you can't find that online. And as I think I mentioned before, this is referred to a delinked payment model or a subscription-based contract, but it's also often referred to in the kind of grey literature as a Netflix model. So you could compare this to your Netflix membership or your gym membership. You pay the same amount for those regardless of whether you go to the gym 5 days a week or 5 times a year. It's that sort of concept. So I'll next just take you through a bit of the technical detail of how our approach was different from the more traditional health technology assessment approach. For those of you that are familiar with our normal methods, I'll try to keep it not too full of jargon. So how does the antimicrobial evaluation differ from our normal approach to technology appraisal, HTA? So we're asking a fundamentally different question here. So normally NICE's advisory committees are tasked to make a recommendation when they're looking at individual medicines and interventions and treatments, make a recommendation that either yes, it should be used because it's clinically and cost effective compared to current practice, or no, it shouldn't because it's not, or maybe yes for a subgroup of patients where it's cost effective. That's based on a price proposed by the manufacturer. So the per pack price is taken into account. Whereas for the antimicrobial evaluations, the two that we've done already in the pilot, and going forward, the committee won't be asked to make a binary yes or no decision. Instead, they're concluding on what the value of the drug is to patients in the NHS. And I'll come on to how we define value in a minute. And it's that conclusion that informed commercial discussions to agree the fixed annual fee. Now, the value that NICE estimate isn't a monetary value, but rather the value in what we call quality adjusted life years. So those familiar with our work may be familiar with that as a unit of measurement that NICE use. So a quality adjusted life year is a way of thinking about not only how long a person lives, or how many extra years of life a new treatment will give them, but also thinking about the quality of those remaining years of life. So one QALY is equal to 1 year of life in perfect health. If you had a year of life, but your quality of life was, say, 80% of the average person, then the QALY would be 0.8. And again, if you're familiar with NICE's work, you'll know our usual modelling output is the incremental cost-effectiveness ratio, the ISA, and that's the basis of our recommendations. So the ISA means the cost per QALY gained with the new treatment. But for antimicrobials, we're not factoring in the cost of that packet of drugs because the companies won't be getting paid per packet. So we're simply thinking about the benefit of this treatment in QALYs, and NHS England use that to calculate what the fixed fee per year should be. The QALY estimate is also called the incremental net health benefit. And in terms of how we actually measure that and capture that, how do we work out what that is? Well, like with any other health technology assessment, the direct health effects to the patients treated are important, of course, and we capture those. So the clinical benefits in terms of improved survival, improved quality of life, fewer side effects, reduced time in hospital, all of those things that we would normally look at. But to recognise the value of antimicrobials, we also consider other benefits that we don't normally capture. So first of all, benefits to the person treated, the person with the infection. So as an example, if you took a person that had cystic fibrosis who needed a lung transplant, but they had a chronic drug-resistant infection, making them too unwell to have that transplant. Well, if the new antimicrobial makes them well enough to go on and have that transplant, we factor in the health benefits of that. And we also capture in the wider public health benefits to people who aren't even ill with the infection, who aren't treated with the new drug. So other people in the hospital who we don't want to catch this bug, other people in the hospital whose operation would be cancelled if there was an outbreak, if this superbug in the ward had to be shut down, or simply they can't share doctors and nurses between infected and uninfected patients. So these other benefits, and that they were just a couple of examples, which we don't normally capture, that we captured in the pilots and we aim to do going forward, are often referred to as the STEADY values. That's an acronym, which I'll define on the next slide, please. I should note these are all still health related. We're not looking at broader benefits like productivity losses in the workplace and that kind of thing. So we're still focusing on health. So a bit more about the STEADY values. As you can see, it's the acronym for the words in bold on the slide there. So first of all, we have spectrum value. You may hear people talk about the spectrum of an antibiotic. So spectrum value refers to the benefit of a new, narrower spectrum antimicrobial. So that means it's active against fewer very specific bugs and resistance mechanisms, replacing broader spectrum antimicrobials, which reduces problems of resistance associated with their use. So a broad spectrum antimicrobial is active against lots of different bugs and resistance mechanisms, which is great if you don't know what's causing the infection and you want to increase your chance of killing it while you're waiting for test results, or you just don't know, but they're not good because they do drive resistance. Transmission value was maybe more obvious, so that's about reducing the spread of infection. Enablement value, I alluded to just before with the examples I gave. That's about the benefits of being able to perform medical procedures that wouldn't otherwise have happened if the infection was not properly treated. So again, this can be, as I mentioned before, the person with the infection or other people in the hospital. Diversity value, I alluded to this at the beginning, refers to the benefits that introducing new treatments into the formulary offers, by diversifying the range of treatments available, thereby reducing the use of individual treatments and reducing the selection pressure on those treatments to reduce resistance development. And then finally, the I of study is insurance value. So that refers to the benefits of having a new antimicrobial under lock and key, being reserved until you really need it, when there's no effective treatment left. Think about the insurance premiums you pay to protect your house against fire, theft, damage. There's a prominent person in the antimicrobial resistance field called John Rex, and he describes antimicrobials as the fire extinguishers of medicine, and I quite like that analogy. So the study values are really, really key to this work and to estimating and recognizing the full value of an antimicrobial. And it was, if you take a look at our guidance, or if you've seen or you even observed the committee deliberations or read the economic report, it was really hard to quantify these in the evaluations, but NICE is actually the first HTA organisation anywhere in the world to attempt to quantitatively estimate the full benefits of an antimicrobial in this way. So some really pioneering work was done here. Next slide, please. So what's next? As I said, the work doesn't end with our pilots. We're committed to implementing this new evaluation and payment approach routinely to other antimicrobials. We and our stakeholders identified a few challenges with replicating exactly the approach we took in the pilots, mostly on the evaluation side, the economic modelling side. So immediately after the pilot concluded and these payments started in July, we ran several workshops to get feedback from internal and external stakeholders, including industry, academia, key opinion leaders in AMR. And we're now fine-tuning and revising our methods to make it fit for routine use. So we're continuing to engage with those stakeholders in the lead up to Christmas, and we'll put a detailed plan into the public domain, so you'll all be able to see it and comment on it. So we'll be consulting on the new approach early next year, aiming to start routinely using it next spring, I think. Next slide, please. This is my penultimate slide, just to highlight that a key part of our next steps is to encourage similar incentives in other countries. As I said, NICE is the first to do this anywhere in the world. The first quantification of the value of really important antimicrobials for superbugs, which are designated as a global priority by the WHO. But England, so I should mention this, the pilot schemes apply only to England, but our future kind of routine arrangements will apply across the whole of the UK. But these contracts are in force in England only at the moment. And England represents only 3% of the global antimicrobial market. So it's no good us doing this on our own. Companies won't be incentivized to spend lots of money researching and developing antimicrobials if they only recoup 3% of their cost. So we need the rest of the world to pay their so-called fair share. And that's a terminology that is used a lot when talking about pull incentives for antimicrobial resistance, is fair share payments. And there's a dedicated team in our Department of Health and Social Care that have been engaging with other nations throughout the project and in designing the project and all the way through and continue to do so. So they've been sharing learnings and asking for feedback on this work at meetings with the G7 and the G20 countries, and are also engaging closely with the US and Canada. And the team contributed up thoughts to the draft bill in the US called the Pasteur Act, which is looking to do a similar thing. So, global collaboration is really important here, of course. And then for my final slide, just to end, I've included NICE's webpage, which has everything you need to know about this work. So the very detailed methods we used in the pilot, all of the clinical and economic evidence we considered, the actual guidance documents published by us as a result of the committee meetings, a copy of the contract between the two companies and NHS England. The actual payment levels, which are confidential, are removed, but all of the other contractual elements are there to see. Most recently this week, we published what we call a lessons learned report. So that's a summary of the reflections and feedback from ourselves and all of our stakeholders that we've been collating over the summer, and some ideas for how we move forward. And I mentioned the consultation on our detailed plans for the routine process early next year. So this website will have details of that consultation. So watch this space essentially. And that's it from me. Thank you.
Okay, thank you, Sophie. Very interesting presentation. Thank you, both speakers. So it's time to the questions. The question for both speakers. I think that we have time for 3 or 4 questions. So I am reviewing the chat. Okay, so the first question for Francesca. Francesca, you mentioned that protonamide is not in the guidelines yet. It seems there is a gap between drug development and policy guidelines. How do you feel research consortium like ERA4TB, who are developing new treatments and treatment regimens, should be working to change policy? For example, should they be liaising more with national TB programs to involve them in early drug development?
Thanks for that question. And I think the answer is simply yes. You wouldn't bring to market anything without asking the end users. And although the end users are patients, the kind of gatekeepers are the national TB programs. So as a guess, what would the national TB programs ask for? The first thing is shorter treatment. So treatment that doesn't take 6 months, that can be completed within 4, 2, or even shorter months. The next thing is that the need for monitoring. Bedaquiline came with a black box warning that says that there could be QT prolongation that caused unexplained deaths, which means that all national TB programs have to do, every 2 weeks, have to do an ECG on patients. That may sound strange to you, but as mentioned before, in South Africa, we're having load shedding, which means that we have interruptions to our power supply 2 to 4 hours every day. So that has, so asking people to do monitoring like ECGs or blood tests comes at a cost. Can we get a cleaner drug with fewer side effects? But I think overall, the quicker we can get treatment over and done with, and patients can be cured with the low chance of relapse, the better. So convene forums with NTP managers. What do they want? Before, not necessarily, we can't take everything into consideration, but some of them we can. Thanks, Alberto.
Thank you, Francesca. So another question, this one for Sophie. Are the steady values value set that NICE introduced it specifically for the AMR work?
In short, yes. There's been quite a bit of literature. I think originally a report from the Office of Health Economics, which we reference on our website somewhere, and also the report that I put onto the slides from EPRO, defining these and recognizing these is really important for understanding and capturing and rewarding the full value of antimicrobials.
Okay, thank you, Sophie. Okay, the next question is for Francesca. So Francesca, what do you think there are the priorities for tuberculosis research? For example, should it be finding new treatment for drug-resistant tuberculosis or non-resistant tuberculosis, or perhaps drugs for pregnant women with TB, or maybe the focus should be on reducing side effects? Please, Francesca.
So the answer is yes to all of the above. I think that what I have observed in my career is that we normally get antimicrobials that work in the worst possible situations, multi-drug resistant organisms. And then they kind of drift down as we find them to have less side effects into more common usage. So there is a thinking that we should have a pan-TB regimen, that a single regimen would treat drug-resistant and drug-sensitive TB. Personally, I think that that's a bit of a pipe dream. If we ever go down that route, then what we need to do is to make sure that we keep an eye on the development of resistance within the communities that we serve. We need, pregnant women seem to be protected from research or withheld from research, despite the fact that 200,000 of them will get TB in the next year. So once a drug has been shown to be safe and efficacious, PK studies should be done in pregnant women because pregnant women's metabolism is different from non-pregnant women. But I think that the key thing is we want to be able to treat TB in a shorter period of time than 6 months. And COVID, if it taught us nothing, the one thing that it did teach us is to get people out of the healthcare system and back to good health as quickly as possible. Thank you.
Thank you, Francesca. The next one is to Sophie. Okay, how do you gain data about genuine quality of life? What is the definition or definitions of quality of life?
Great question. So from NICE's perspective, there are lots of questionnaires to capture quality of life, patient-reported outcomes measures, PROMs, they're abbreviated to. There are disease-specific ones, there are generic ones. I could probably talk for an hour about that alone. But for NICE, we rely on, to make sure we have consistent decisions across all of the different interventions that we look at in healthcare, in social care. We need, to ensure that consistency, we need to rely on one instrument to capture quality of life, and so it has to be generic, not disease specific, capturing quality of life regardless of what's affecting it. So we prefer the use of an instrument called the EQ5D. It's a questionnaire with 5 questions about generic areas of your health and quality of life: mobility, pain, anxiety and depression, self-care, usual activities. So that's how we capture it. There are ways that you can use other questionnaires in clinical trials and then work out what the equivalent number on an EQ5D questionnaire would look like. But there are questionnaires that are used in clinical trials, and that's sort of our gold standard. Our number one is a questionnaire in a clinical trial, the EQ5D. There are lots of other options if that isn't available. For some of these really serious infections that we're looking at, that can be quite hard to capture. Some of the infections that we were dealing with in the pilot evaluations, you're in critical care, you're in an intensive care unit, you're not answering questionnaires. But there are ways around that, but hopefully that helps to answer your question.
Okay, thank you, Sophie. We have more questions in the chat. The next one to Francesca. Francesca, have you investigated how improving access to clean water and effective waste system may improve health, so each person will be much healthier and robust, so less vulnerable to an infection?
No, because that's outside my area of expertise. I'm a TB doctor, but I agree. If every person who lived on this earth had access to clean drinking water, access to family planning, and access to enough food, that the impact of TB would be less felt. But I don't have numbers to quantify that.
Thank you, Francesca. Next one to Sophie. Sophie, please, could you expand on what it means by fair share? How is calculated and used? how does this create pool incentives?
Yeah, sure.
Thank
So am I unmuted? Yes. So a country's fair share is their proportion of the global pull incentive for a new antimicrobial, and I'll kind of break that down a little bit. So that's the payment to the company that would ensure a sufficient return on their investment to make the development of a new antimicrobial worthwhile. And the way in individual countries, I mean, there's lots of literature been published. I can share it afterwards. Kevin Outerson has done lots of work on this, and there's been several papers before him, but he published something most recently, I think this year, that suggested what the global size of a pull incentive in monetary terms would be for the pharmaceutical market to recoup their costs. So then an individual country's fair share is calculated by rescaling that to account for the country's, proportion of the antimicrobial market, GDP, various other things. So yeah, I can share some literature if that helps.
Okay, thank you, Sophie. We have the last question to Francesca, and we have also another comment. I think that I can read the comment for Josephine. Just a comment on the treatment of pregnant women. Another IMI-funded project called Conception is trying to reach that knowledge gap, developing infrastructure for medicine safety in pregnancy and breastfeeding. Okay, thank you, Josephine. It's very interesting. So the question to Francesca. Francesca, would you advise on including in trials patient stratification by severity and diverse study arm, for example, treatment duration? So less ill patients could benefit from shorter treatment, for example?
Advise it. Although TB trials are very expensive, and they take a long time to conduct, the methods that we have are at the moment of deciding on disease severity is the smear grading. So whether it's negative, positive, 1+, 2+, 3+, pretty gross tool and largely dependent on how well a patient can cough. Chest X-ray findings, BMI, and then finally, as I'm a South African doctor, HIV positivity or not, and baseline CD4 count. So I think that it would make sense to stratify upfront so that we enrich for the population who either will do poorly or will do worse, and we can have better bang for our buck in terms of what we get out of clinical trials. Thanks, Alberta.
Okay, thank you, Francesca. So to wrap up this first session, I wanted to thank all you again, all the participants, the speakers, our host, NICE, ERA4TV, for joining and participating in this webinar. I think that it has been a very interesting session, and I look forward to the next ERA4TV webinar that probably will be happening in March 2023 to coincide with the World Tuberculosis Day. So once again, thank you everyone and have a nice day. Goodbye.